Hello everyone, good morning, Assalamu alaikum. Alright, today I will make another video which continues my last video about Biophysics Part 1, and now we are on the second part of the Biophysics explanation, which will cover my research interest in bio and robotics. So I will divide this video into 3 sessions. In session 1, we can learn some facts about bioelectromagnetism. And for the second part, we will discuss my research project. And for the last session, we will learn some interesting facts about bio-inspired robots. Okay, let's begin. Bioelectromagnetism is one of the course I had taken in my master degree. Thanks to Professor Jaco Malmifu and Robert Plancy for this amazing book, which has inspired me a lot and helped me with my research. And as you can see here, there are several human organs illustration. And there are 3 columns that explain the segmentation between electricity and magnetism in relation to bio terms. So after saying this, we all know that these human organs have a correlation with electricity and magnetism. For example, the brain and muscle have a certain activation process that involves electrolyte ions, which can be measured by a specific medical device. And it is a similar way that happens in the heart and other organs. We can see here in the middle a diagram that explains the multidisciplinary science and engineering subject that make a new terms. When combined, the subjects are interacting with each other and make several meaning like bioengineering, biophysics, bioelectromagnetism, medical physics, medical engineering, and medical electronics. And we can see here bioelectromagnetism is intersecting with every subject. And next, play the video.
But squid are not vertebrates. They are allied to the sedentary seashells of our shores and belong to a far older group of animals, the cephalopods, whose ancestors once dominated the ancient Paleozoic and Mesozoic seas. In modern forms, the lower half of the body consists almost entirely of the mantle cavity, and it is the contraction of its muscular wall which expels seawater out through the funnel or siphon just below the head. A 350-gram squid can squirt about 200 milliliters of water, about half a pint, in less than a fifth of a second. Opening the mantle cavity in the mid-ventral line exposes the squid's true body wall, through which the viscera can be seen. This is called the stellate ganglion, and when the gill is removed, the reason is clear. Numerous nerves just below the skin radiate from it in a star-like pattern. But each of these tubular structures must be a single nerve fibre, a giant axon formed by the fusion of very many smaller ones. Young confirmed this hypothesis when, using a pair of simple electrodes, he stimulated a stellar nerve. Large contractions of that part of the mantle served by the stellar nerve were only obtained if the tubular structure within it remained intact. The change is only produced, though, if the stimulus is large enough because, like all single nerve fibres, the giant axon has an all or none response. This is the action potential.
In this lesson, we will take a much closer look at the mechanisms by which an action potential is generated at the axon initial segment. There are three key events. First, at that point in time when the threshold of excitation is surpassed, the sodium channels open and sodium ions rush into the cell through the membrane at the axon initial segment. This is the beginning of the rising phase of the action potential. Second, as the membrane potential is depolarizing due to the influx of sodium ions, the voltage-gated potassium channels open and potassium begins to leak out of the cell. Third, once the membrane potential depolarizes sufficiently, the sodium channels become inactivated, stopping the sodium from leaving the cell. This is the end of the rising phase of the action potential. But the potassium channels are still open, so potassium continues to leave through the membrane. This is the beginning of the repolarization phase of the action potential. Potassium continues to leave through the membrane until such a time that the cell is repolarized and the potassium channels begin to close. The closing of the potassium channels marks the end of the repolarization phase of the action potential. But the potassium channels close rather slowly, so the membrane potential becomes hyperpolarized, and that hyperpolarization is maintained for a period of time until the membrane returns to the resting membrane potential.
The area of axon that fired an action potential a millisecond ago cannot become depolarized now. It is refractory because its sodium channels are inactivated. In this way, the action potential propagates in only one direction, like a fuse. Now, 2 milliseconds later, the positive charge spreads to the next adjacent region of the axon, and it reaches its threshold and fires an action potential. The region of axon that fired a millisecond ago cannot be depolarized. It is refractory. The area of the membrane that fired 2 milliseconds ago has now returned to its resting potential, but it is too far away to be influenced by the current action potential. In this way, the action potential migrates, like the fire on a fuse, toward the axon terminal.
So in order to show that, we have to get some vocabulary out of the way. So I'm going to do what we do in the classroom with students. We're going to do an experiment on electrophysiology, which is the recording of the body's electrical signals, either coming from neurons or from muscles. And I'm putting some electrodes here on my wrists, and as I hook them up, we're going to be able to see a signal on the screen here. And this signal may be familiar to you. It's called the EKG, or the electrocardiogram, and this is coming from neurons in my heart that are firing what's called action potentials. Potential meaning voltage, and action meaning it moves quickly up and down, which causes my heart to fire, which then causes the signal that you see here. We're going to record the electrical potential from this plant right here, this mimosa. And so what we're going to do is I've got a wire wrapped around the stem, and I've got the ground electrode where? In the ground. It's an electrical engineering joke. All right. So all right. So I'm going to go ahead and tap the leaf here, and I want you to look at the electrical recording that we're going to see inside the plant. Whoa, it's so big. I got to scale it down. All right. So what is that? That is an action potential that's happening inside the plant. Why was it happening? Because it wanted to move, right? And so when I hit the touch receptors, it sent a voltage all the way down to the end of the stem, which caused it to move. And now in our arms, we would move our muscles, but in the plant doesn't have muscles. What it has are water inside the cells, and when the voltage hits it, it opens up, releases the water, changes the shape of the cells, and the leaf falls. And now here's my Venus flytrap, and inside the leaf you're going to notice that there are 3 little hairs here, and those are trigger hairs. And so when a fly lands, I'm going to touch one of the hairs right now. Ready? 1, 2, 3. What do we get? We get a beautiful action potential. However, the flytrap doesn't close. I'm going to touch the Venus flytrap again. I've been talking for more than 20 seconds. So we can see what happens when I touch the hair a second time. So what do we get? We get a second action potential, but again, the leaf doesn't close. So now if I go back in there, and then if I'm a fly moving around, I'm going to be touching the leaf a few times. I'm going to go and brush it a few times. And immediately, the fly trap closes. And so what would happen if we took the action potential from the Venus flytrap and sense it into all the stems of the mimosa. We should be able to create the behavior of the mimosas without actually touching it ourselves. And so, if you allow me, I'm going to go ahead and trigger this mimosa right now by touching on the hairs of the Venus flytrap. So we're going to send information about touch from one plant to another. So there you see it. So, so I hope you learned a little bit something about plants today. Not only that, you learned that plants can be used to help teach neuroscience and bring along the neuro revolution. Thank you.
Okay, from previous video, we have learned that electrical properties like action potentials in membrane cells can be measured by a specific device or electrodes. This is Sir Alan Hodgkin and Andrew Huxley, two of the three Nobel Prize winners in physiology and medicine, 1963. And this is their equation, Hodgkin-Huxley equations, about ions conductance that correspond to membrane potential voltage. The excitable cells include neurons, muscle cells, beta cells of the pancreas, pulvinules in Mimosa pudica, and other cells in plants. From the excitable tissue phenomenon, psychologist Frank Rosenblatt in 1958 developed the perceptron, the first artificial intelligence machine that can learn and make decisions using a similar concept to neurons in the human brain. This concept is proposed as a bridge between biophysics and psychology. And now, in the modern world, we have more advanced technology in artificial intelligence because of that basic discovery. Now we got the second session, where I would like to tell more about biophysics according to my research project. Biomimetic is a research area that studies the phenomena and process of nature with the aim of understanding them and then being able to use and modify their mechanism for our benefit. We can see here several biomimetic examples like biomimetic materials, biomimetic robots, inspired by animal, plants, and even human movement mechanism. Mimosa pudica, or sensitive plant, or also called touch me not plant, is well known for its rapid plant movement. This plant has several nastic movements like thigmonasty, seismonasty, photonasty, and nyctinastic movement. The thigmonastic movement is shown by its reaction when the leaf is touched and will perform a closed leaf reaction. So what happened in mimosa plant is the same concept since it has an excitability phenomenon in their cells. As you can see here, it's the cross-section of the Mimosa pudica plant, and that happens in their phloem tissue, where those ions exchange and propagated electrical signals or action potentials. So this is my research project. This is my Mimosa pudica biomimetic robot, consisting of 3D printed parts, touch sensors, servo, accelerometers, integrated circuits, resistor, capacitor, and microcontrollers. I made two different robot models here which have different activation methods using different microcontrollers. The first model of this robot used a local activation and controls with 8 artificial electronic excitable tissue. As you can see here, this little PCB with IC chips on the board. It is the electronic excitable tissue that can fire action potential signals after reading the sensor voltage input when stimulus reaches the given voltage threshold. So when three nodes of the leaf sensor are touched at the same time, the robot will fire the electrical analog signal to its nearest neighbor. You can see the picture below of the oscilloscope measurement. There are 8 signals representing the leaf electrical signal and movement activation of 8 leaf nodes. The timing is programmed into a 80Tiny85 microcontroller according to an actual video of a mechanical stimulus experiment from a real plant sample with the same touch leaf position. For the second model, the robot sensors and actuators are centralized into one microcontroller, which is Arduino Mega. So we have the dataset of the input sensor location combination with 2 or 3 nodes that touch at the same time. We will have the unique input combination which will be distinguished from each other. For example, we have 3 data here, S1 until S8 representing the sensor SN location. At the end node, the RS means the resting state condition, 1 for a full time recovery which is 20 minutes after the last reaction, and 0 is for 10 minutes resting after the last reaction, which is just enough for the mimosa plant sample to reposition their leaflets back. The real experiments provide 26 location combination. So from this table we can create the input-output condition where we can train and use the neural network model in the robot decision making process. And will be executed by a computer. We can see in the video how it works. And what's the potential application of these research projects? We can see here there is a solar cells that can be mounted on this robot structure. So this adaptive structure can be applied to a real solar panel to reduce the damage risk and even lower the cost of the maintenance or cleaning the solar panels because it is protected from dust or sand while it's not harvesting energy. This concept can be developed more. For example, we can use it in the satellite or space station, which can be damaged by space objects like asteroid, space junk, and space debris. And it's not only that. From this research, I hope it will be inspired the adaptive structure of other plants that have not been discovered in other rapid movement plants. For example, like Nastic movement, tropism movement, and taxis movement. Venus flytrap biomimetic robot was invented in 2011 by Mohsen Sahinpur, and there are many plants which still available for us to build the next research project, like sundew or drosera plant, dancing plant, and many more. And the technology which can be used in the biomimetics application is very fast. Because my research is talk about electrical device panels like solar cells, so I present several pictures here about the James Webb Space Telescope, which was already damaged because of space objects. So I think it makes sense to apply my research content to this kind of structure. And then we already have adaptive structure like ISS, origami satellite, and foldable flower-shaped solar panel. Bio-inspired robots are sometimes called bionic robots or biomimetic robots. We can browse YouTube videos about that terms and we will get some cool robots like Festo bionic robots, the popular MIT cheetah robots, and later which became viral, this madman built a robot suit for snakes. There are also companies working with exoskeleton robots that will help people with disability. Harvard University also built some cool robots like play robots and swarm robots. In the soft robotics area, usually there is a lot of research about octopus arm or tentacles to build tentacle robots. And there is also gecko robots. Of course, the most incredible bio-inspired robot video is win by Boston Dynamics. They stunned the world with advanced robotics in a very magnificent way. Literally, it is a role model to the other robot companies or developers. Okay, that's all for this video. I hope I can make more videos with interesting facts about biophysics, bioengineering, and bioelectromagnetism. So don't forget to press the like button, subscribe, and share this video with your friends. Stay positive and always curious. Bye!